So DNA is made up of 4 bases, nucleotides: adenine, thymine, guanine, and cytosine. And these can attach to each other in a temporary manner. Now, RNA is different. And one of the major differences is that although in DNA, adenine and thymine attach to each other, in RNA, adenine will attach to uracil. So if the DNA molecule has an adenine, the RNA molecule will have a uracil. Minor change, but nevertheless, it's important in terms of how RNA functions. Now, messenger RNA, once manufactured, can leave the nucleus, which DNA is just too big to do. So it can leave the nucleus and go out into the cytoplasm, the basically water-like fluid that surrounds the nucleus and that's in the cell. There, it goes to an organelle that's called a ribosome. We probably have millions and millions of ribosomes in our cells. And the ribosome's job is to read the message on the messenger RNA. And it reads it in a code of 3 letters. So 3 nucleotides make up that code. And those 3 letters code for 1 amino acid. And usually they code for only 1. We have approximately 20 amino acids, but usually it codes for only 1. Sometimes it may code for 1 of 2 amino acids. But those 3 letters code for an amino acid. How this works is the ribosome reads the code. It knows where to start on the messenger RNA. I shouldn't use the word know, but it chemically can identify where to start, and then it moves down that 3 letters by 3 letters by 3 letters, and it calls in a molecule called transfer RNA. And transfer RNAs are specific for each of the amino acids. So if there's a 3-letter code, let's say the 3-letter code on messenger RNA is CGA, the code on transfer RNA would be GCU, and that would carry a specific amino acid to it that transfer RNA gloms on to the messenger RNA within the ribosome. And then the next site is read, and that transfer RNA gloms on, and then the 2 adjacent amino acids bond to each other, and the transfer RNAs leave, recycle, pick up another amino acid, and then the protein grows. So you have a protein that grows, may change in shape, and that's how protein is manufactured. So when that protein is manufactured, that gene is expressed. But life isn't that simple. Science is never simple, because expression of the gene is one thing, but in fact, there are things that can modify genetic expression. So let's go back to that messenger RNA molecule. That messenger RNA molecule that's manufactured in the nucleus has 2 different kinds of areas in it. And those areas are pulled straight out of the DNA. All right? Area number 1, type of area number 1 is called an exon. And an exon is going to be the part of the DNA code that will be expressed in a protein. The other parts are called introns. And for years and years and years, they said, oh, introns were garbage, you cut it out, it's gone. And what happens is that there are enzymes that cut out the introns, and then the exons get to move together and attach each other, and then you have the real messenger RNA. And that is what's ready to be read at the ribosome. So what happens to the introns? They're not garbage. The introns, in fact, move out of the nucleus into the cytoplasm, where they end up through, again, enzymatic reactions, becoming RNAi. So these things are called microRNAs, and they become RNAi, which are considered inhibitory RNAs. What do? Well, there's your ribosome, the ribosome sitting, reading the messenger RNA, and they can attach to the messenger RNA and basically disrupt this process, which by the way is called translation, where you make the mRNA code into a protein, they disrupt it. And therefore, they suppress expression. So a protein suddenly that should be expressed isn't expressed, but it's not expressed in the cytoplasm as opposed to in the nucleus.